All right. So the goal is to make a figure that goes with this. So okay. So we have a start out with a scientist. Scientist in PPE. Scientist with PPE. Business suit. Okay. Scientist starts. Should I do this or should I go with more? I don't know. I like the. So this is him. Insert text. No some. Worker. No PPE. Full PPE. No symptoms. Lineage B infected. This is SARS-CoV2 lab leak. Actual. Okay. Accidentally infected with lineage B. Full PPE. Then this person travels. Pretty direct travel. Takes a car. Drives. It has to be a car. Basically. All right, and this person drives immediately. Drives. No. Directly. Okay. This goes to the seafood market. This person. So we are talking about, we're making a figure. If it is true, if the lab leak is true, what would actually have to happen in order for this to contend with all of the available evidence. And that's what we're talking about here. So I have to drive directly to the Huanan Seafood Market. One of the least trafficked places. Huanan or the Hunan? I think it's the Hunan. Oh, no, it's the Huanan. That's right. Oh, add to personal dictionary. Low. So. Okay, let's go put this here. So. And then we want to put in low trafficked. Least. And then once there, infects multiple. Let's see here. We're trying to put in like multiple. So it's like this. We're going to put it into a bunch of different places. Okay. Raccoon. Oh my god, they have raccoon dogs here. That's so cool. All right, raccoon dogs. That's so funny. Okay. What were all the places that we actually found samples? Let's see. What did they find samples with? Trying to look here. I think they found. Here we go. Lots of raccoon dog. Weasels. Siberian weasels was another one. Okay. So weasels. Raccoon dogs. Delete. This, I'm going to put raccoon dogs here. Raccoon dog. Okay. And like a weasel. There we go. A ferret or something works. This is a ferret. Siberian weasel. Mass palm civets. It kind of looks like a ferret. Siberian weasels. What do you look like? Okay. Let's put Siberian weasels here. I think that's going to be fine. Yeah, there's no weasels here. Okay, Siberian weasel. go. Be accurate. Okay. Boom, boom, boom. This. Well, oh, okay, that's cool. This. Okay. 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 So everything that we know with the scientific evidence given to us, an employee at the Wuhan Institute of Virology accidentally infected with lineage B, despite safety measures, immediately went into one of the least traffic markets, drives directly, moves directly, no other contact. I don't know if we could say drives. Okay. And all the people near it deliberately spits, coughs. Okay. And into animal cages. See an animal cage here. Rodent feeder. Skinner box. Okay. Let's see. And other people. Let's do like a crowd of people. No. People, person. Group of people. Oh, that's cool. Okay. Directly, no contact. Let's move this up. Here, we're running out of space. I think I'm gonna have to put this over here. This is the one on seafood market. Low traffic. Here, 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 up. Okay. And then once there, I like to group this together. Group. This and this together. Group. Okay. Okay, and then once there, let's repeat this, transform it, flip it horizontally. There we go. So now this, let's use there. This spits. Okay, spits and coughs on raccoon dogs. This raccoon dogs, Siberian weasels, the crowd there. I think this is a little bit smaller. Raccoon dog a little bit. Oops. Raccoon dog a little bit smaller here. Okay. Siberian weasel. It's just a small, touch smaller. Okay. And then the crowd. So this is the crowd of people. It's the same size as everything else. Okay. And then this is just the other random crowd of people. Do, 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 do. Gonna bring this to the front. Bring forward. Arrange. This is the crowd. S west plus east sides. So this is the people in west and east sides of market. Okay, this. Okay. There. Okay. So this is how it had to happen. And then employee A. Okay. And then shapes, arrows. to do this, and then do this, this. 1 week later, and then fold this. One week later. Just go to the top. This. Group. Nice. Boom. Boom. Okay. Now this is lineage employee B. Lineage A. And this person does the exact same thing. This is what happened. This is what would need to happen. This is what would have to happen. Okay. Let's make some red onto this. I like the idea. Let's see more. This. Okay, cool. So a different employee there. There you go. Cool. So this is what would have had to happen. This is, we'll make this blue. Cool. This is what lab leak would have to happen. Cages. Cage. I need a cage. Cages. Animal cages. Okay, this group. That one group. Text, animal cages. All right, this is it. Okay. Okay. And then now let's make a copy of this. Do copy. Okay, so natural. All right. So now we're doing natural origins. No longer this person here. No longer this or this. This. So this could be okay. So we would have what were the ways that we could have, let's see. Wild, zoo, or breeding? So breeding. Breeding center. Okay. Okay. So we have a breeding center. This is how it basically happens. The breeding center that we know, say it's a breeding center that is infected. Biohazardous. Okay. Okay. So we have a breeding center here that's infected. Right? This is infected. Okay, so a breeding center for, let's say, raccoon dogs. So this animal has to go through the breeding center. Okay. Goes from the breeding center to the Huanan market. Okay, so then once in here, now infected. And then once there, then animal cages. And these are infected animal cages. Okay, and then this goes into the lines here of what happened. Boom. People walking by. Leads to infected animal cages. Leads to infects humans. to make it like a virus thing to show that these people are infected now. Okay, so this is it. Okay, so here we go. This is it. This is the natural origins. And then 1 week later, shaboom. Lineage A. All right, cool. So that's the... Here we go. Here's the... This is it. Here's the natural origin. Spit and coughs. Range. Send to the back. You're annoying. Okay, cool. This, and now this. There. Biosafety cabinet. That's a good idea. Biosafety cabinet. Bring to front. It's a biosafety cabinet. Arrange, bring to front. There, there. Okay, let's make this a little bit bigger. Canvas size, a little bit bigger. This, 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 this. I'll make later. Okay, here we go. Okay, here is the new one. Group together. Group together here. Okay. Cool. So this is what would have to happen for lab leak to be real. That's what we're trying to make a figure here, trying to showcase and highlight what would actually have to happen. Okay. And then also this would be natural origins. So it's way simpler. And just one. Here we go. That twice here. Cool. So this is what it would be. So for lab leak to be true, you would need to have a worker inside of a biosafety cabinet that's working the Wuhan Institute of Virology in full PPE, has no symptoms, that has lineage B infected within them, and then never seeks medical care, goes directly, no other contact, to the Huanan seafood market that has low traffic in the city. And then once there, they spit and cough onto animal cages, raccoon dogs, Siberian weasels, and then infects people inside the market. Dang it, why did I do that? There we go. Yeah. So we're trying to figure out how to show, I guess I'm just... Okay, this. So this is part of the paper that we're looking at and we're going to look at. But yeah, this is it. So employee A inside of a biosafety cabinet works for Institute of Virology, has full PPE, no symptoms, with lineage B infected. Later, no antibodies created because the serology tests point that out, that they never have no antibodies created. So this person goes directly, no other contact, to the Huanan seafood market, which has low traffic. And there, spits and coughs on animal cages. This person spits and coughs on animal cages, raccoon dogs, Siberian weasels, and other people, and infects people on the west and the east side of the market. And then 1 week later, another worker inside of a biosafety cabinet, different worker, another on Institute of Virology worker with full PPE, no symptoms, lineage A infected, never seeks medical care, and no antibodies are created. Goes again directly, no other contact, to the Huanan seafood market. Again, that's very low trafficked. And then again, spits on and coughs on people on the east and west sides of the market, Siberian weasels, raccoon dogs, and animal cages. Or infected breeding centers that we know have viral outbreaks of these raccoon dogs, and I can actually just add this. I wonder this. I just added them. Let's see, weasels. Weasels. And then raccoon dogs. Again, inside those cages. Let's bring it to front. again, these are infected. Whoops. This. Okay. I think I'm going to put just the Kuanan market and take out the breeding centers. Because if we're really going to go that far, then we would have to put in the... We'd actually have to put in this whole thing, basically. Which is the whole point. That's kind of why I'm trying to get away from... We're just looking at the outbreak itself. So, therefore, natural origins is actually, I think it's more fair to the actual story of what happened to actually put something in that's actually represents it. Okay. Let's put these on the outside here. So we have that. Put these together. I'm going to take that out. Put my own text here. I'm going to put, what were they? Siberian weasels. Raccoon dog. Okay, and then these people are now walking by or buying. Buying or walking by. Buying or walking by. Whoops, fine, or walking by. There. Okay. Let's see here. Do 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 do. Okay, mitochondrial DNA. Okay. Groups together. Okay, cool. This done. Boom. 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 Cool. I think this is it. Sensors centered a little bit better here. Do 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 do. I'm actually kind of happy with this. Okay. I think that's good. I think that makes sense. Okay. I think that's a good. Cool. I think this is good. I like this. I'm proud of this. Hopefully this better illustrates our frustrations. Cool. All right, cool. Okay. Gonna make it yellow just to make a little bit more distinction. So I have blue and green there. I'd rather have the red be the viral stuff, just for ease, better communication. Okay, this and this, this, this. Is that the same one? No. This. Yes. And this. Nice. Cool. And that's it. I think that's it. Pretty happy with it. Okay. Cool. Okay. Raccoon dog that isn't actually a dog. I mean, sorry, not a raccoon at all. Okay. naming. Okay. I like the spacing on this side a little bit more, so I'm gonna just copy and paste it. All that time for nothing. Here, employee B, green, cyan, green. Yeah. Oh, let's do this. Cyan. I'll do green instead. This. Put this person's lab coat on. That's green. Okay. What? How is this possible? Oh man, this is the stuff that drives you nuts. Okay. There we go. I'm just gonna change the canvas size. There. Okay. Okay, here we go. Boom, 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 boom. Okay, and fix the spacing and okay, let's go. I think maybe clarifying the arrow from the market to the animals to show it contains them rather than it leads to them. Maybe change it to a plus. Ah, that's a good idea. Shapes. I need a plus sign. Plus. Whoops. Okay. This. Oops. Okay, cool. Okay, cool. Cool. I like this. Okay. Let's see. Okay, so the difference being that this one in seafood market plus these infected animals with people walking or buying them infects people on the west or the east sides of the market. And then 1 week later, the same seafood market, lineage A, lineage B, has the Siberian weasels and the raccoon dogs walking or buying. You could walk by or buy. Okay, I think that's more clear. All right, I think that's better. What do you guys think? That good? Is it that good? Pretty good? All right. That's good. I think that's good. Okay. Cool. Exciting. Okay. 60 slides. Oh my god. I don't know if I'm gonna finish this tonight. What's the new SARS-2 Origins intelligence report? This is it. Okay, here is the DNI. June 23rd. Okay, here's another one. Surely, guys, this will be the one. Here's the DNI. Director of National Intelligence. DNI report. June 2023. Change this layout here. Surely this will be the one, guys. This is it. Super short this time. What is this then? Is this not the 2023? Is this the executive summary? The key takeaways. Six. Okay. Where did this report go? Executive summary. This report responds to the COVID Origin Act of 2023, which is called for by the U.S. intelligence to declassify information relating to the potential links between the Wuhan Institute of Virology and the COVID Origins. That's so funny. The National Intelligence Council and 4 other IC agencies assessed that the initial human interaction with SARS-CoV-2 most likely was caused by natural exposure to an infected animal that carried SARS-CoV-2 or a close progenitor. Virus that probably would be 99% similar to SARS-CoV-2. The CIA and other agencies remain unable to determine the precise origin of the COVID-19 pandemic, as both hypotheses rely on significant assumptions or face challenges with conflicting reporting. Almost all intelligence community agencies assess that SARS-CoV-2 was not genetically engineered. Most agencies assess that the SARS-CoV-2 was not laboratory adapted. Some are unable to make a determination. All IC agencies assess that SARS-CoV-2 was not developed as a biological weapon. So it's just like, um, dun 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 dun. Where is the none of these genetic engineering capabilities. I mean, none of these have any evidence. It's very frustrating because like none of these have any evidence of actually what's going on. So I don't know. It's unbelievable to me. It's so crazy. Several Wuhan Institute of Virology researchers fell ill in fall 2019 with symptoms. Some of their symptoms were consistent with, but not diagnostic of COVID-19. The IC continues to assess that this information neither supports nor refutes either hypotheses of the pandemic origin because the researchers' symptoms could have been caused by a number of other diseases. Some of the symptoms were not COVID-19. Consistent with standard practices, those researchers likely completed annual health exams as part of their duties in high containment biosafety laboratories. IC assesses the Wuhan Institute of Virology, maintains blood samples and health records of all the personnel, which are standard procedures in high containment. We have no indications that any of these researchers were hospitalized because of the symptoms consistent in COVID-19. One researcher may have been hospitalized at this time frame for treatment of a non-respiratory condition. They are walking this back. This is unbelievable. I love this. So they were walking back. They are quietly walking back. Improbable. Improbable. Not probable. Not likely. Unprobable. Improbable. Not probable. What's probable actually mean? Oh, what do I do? I don't know if I should put this. It's tough. I'm like making a good faith effort in trying to show how lab leak can actually make how this could happen. But it is extremely unlikely that this is happening. So I feel like I don't want to perpetuate something that I'm trying to show is not probable. Hmm. I'm gonna make one. I'm gonna make a copy and make the whole thing together and see what that looks like. Okay, so let's see here. Let's see if I just Boom. Okay, now this, copy, paste. Oh my God. Okay, I'm gonna group that all together. Beautiful. Beautiful. I want to put this here. Canvas size needs to increase to do with that. Increase a little to do here. Okay. Okay, so this, this, this. Whoops. No. This. Text. Lab leak. Lab leak hypothesis. Lab leak hypothesis. Actual. Natural SARS. All right, I'm trying to figure out how I can best compare these two. If I wanted to compare them side by side. Let's see. I don't know which one I like more. I wonder. Let's see here. Oh, that's money. I think so. I'm basically trying to figure out what I want. This or this? This or this? This or this? Yeah, I think the vertical is better probably. More separation between the two visual would help. Okay, let's try putting this in green and this in red. I wonder. Control to snap. 16. Perfect. Perfect. All right, I'm gonna give equal deference. Equal space. I know people are gonna wonder about that. this. This. Perfect. There. All right. I think that's it. That's cool. I think I do want this to be right there, like the other one. Ungroup this and this. Okay, that and that. Okay. What do you think? Good? Good? What do you think? What do you think? I think this is pretty good. I wonder if I could turn down the transparency here. Like, if I like the. All right. I think that's good. All right. All right. Fixed. Cool. The same size, the same everything. I know everyone's going to care about that. So let's make sure it's all the same. I think that makes sense. Oh, shoot. Is it technically SARS-CoV2? Oh, no. Okay, it is. We want to make sure we got that one. SARS-CoV-2 natural origins. This. This fixed. Beats. Oh, shoot. This needs to be. Export. Highest. Cool. That's cool. I think it looks good. Wait, what the hell? I think the. Damn it, it got thicker. There we go. 1 week later. Dot. Okay, cool. There we go. Cool. Cool. What do you guys think? Good? Good? I think it's good. Okay. Oh my god. I don't know if I want to do this tonight or not. I guess I could try. .44 megabytes. Damn, dude. 30 DPI version. This is COVID Origins 300 DPI. All right. That's pretty cool. That's pretty fun. Okay. I'm going to go get something to drink. 1 second. I'll be right back. Give me 1 second. What? That is so not cool. All right. Hello, hello. Okay. Dun 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 dun. What do you guys think? What Let's see. How should I sign this? Good. Okay, let's see. Sarah. Oh man, of course it's like half off or whatever. It always happens that way, doesn't it? Allow. Download. Where's number 2? Cool. Do I want to sign it? You know what I mean? I feel like I have to. Just in case it does. Yes. Accept. Yes. Close. Let's see here. Okay. Let's go. Wonder. Oh my god. Oh my god. Oh my god, man. Damn. Fucking Twitter, dude. Jesus, that's depressing. I can't even. Oh. All right. Okay. Here we go. Here we go. I don't know how to sign this. I don't know what to do. How do I put my name on this without just shitting on it? Putting like my name right here. The peer.review. Source. The peer.review. Is that good? Is that good? That be it? Let's see. Source. The peer.review. I like Ramato. Oh, that's what everyone else is using though. Source Code Pro. Roboto Mono. That looks cool. Merriweather Linux. Times New Roman. Helvetica. Oh, that's a good one. That's a classic. That's one of my favorites. Does that make sense? People like this? Mm. I love it. Thinks I want to do it like. That's so weird. So funny. I actually don't care about that. That is so weird. Check that. There we go. That's good. And then this. 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 Why do you hate me? I don't know if this is good. I don't know, guys. There you go. I think that makes sense. I hate it. I hate it, but it also balances out on both sides. I think I'm gonna make it way smaller. I actually like the side. I don't know. No, I'll put it in the side of the. Okay, I think that's good. Source centered, probably. All right, I like that. Okay. All right. That's weird. They're not lining up perfectly. That is weird. Why? There. 3, 3. Okay, perfect. That is good. Okay. Cool. Okay. Dude, do I put my logo in there too? This is what it would look like. I did that. My little mountain. Um, I don't know. How do I just like remove the background of this? Ugh. Can I photo? Is there layers that I can just like? That'd be legit. This. Wait, weird. There is a background. How do I just flood the... That's dope. And then can I just remove the background? Erase brush tool. Background erase. Whoa. Flood erase tool. Yeah. Sick. Whoa. I just did something. Yo. Live. Just did that. Just did that live. Okay. That's cool. 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 Logos. Affinity file. Okay. And then I'm going to export. Transparent. Dope. That looks legit amazing. Okay. That's my favorite logo. I did that. Everyone. How fun. Okay. Export. Man, I hate putting my logo stuff. It feels so weird. It feels scummy. Why does it feel scummy? Okay. All right, here it is. 600 DPI final. This is okay. COVID Origins 600 DPI. Okay. Hey. Oh, Dr. Bwin, how's it going? Dude, you are here for a really cool thing that I'm doing. So I am in the midst of basically compiling 2 lectures that I did. I was an adjunct professor for microbiology, and I taught a class, and I had 3 lectures at the end that were open, and I could do whatever I wanted. So I wanted to talk about the biggest microbiology event that everyone's gone through. It's just the pandemic, and basically have 2 hours of of presentation I need to go through. But at the end here, I'm basically giving this scenario where what would have had to happen if the lab leak hypothesis was true. And what I'm doing here is actually making a figure so that people can understand kind of like my argument, in good faith. I'm like saying like this, it is still highly unlikely, right, that lab leak is true, but in the case that it is true, what would actually have to happen? And that's what I'm doing here. So I am putting together a figure. And this is like my first time. I've never tried this type of content before on YouTube. I've done me doing research. I've done me doing kind of like an editorial opinion piece. But I've never synthesized multiple papers. I have about 8 papers here that I'm kind of synthesizing down into this presentation. Or it's technically a lecture. It's like what I would be lecturing to the microbiology class. And this is it. So this is the culmination. This is the final kind of hurrah. So this is the ending of the presentation, where I'm putting it all together and kind of like showing what's going on. Yeah, exactly. So this is what would have happened. I mean, every good science theory has to encompass previous data, right? So that's what this is. So basically, for the lab leak conspiracy to be real, you'd have to have an employee that's working at the Wuhan Institute of Virology wearing full PPE, working in a biosafety cabinet, among other precautions, with lineage B specifically. So it's a genetically distinct strain. And we know this from a paper that shows this is Picard 2021 that shows that genetically there's actually 2 lineages that actually outbreaked at the same time. Well, actually not at the same time. Lineage B was first, and then a week later was lineage A. So we kind of know that from sequencing now. But the point is that this person never seeks medical care, and their blood was later tested and had no antibodies for SARS-CoV-2. Then he goes directly, with no other contact and no other place, to a low-trafficked Huanan seafood market. And then once there, this person has to spit and cough onto animal cages, on raccoon dogs, in Siberian weasels' mouths, and then has to proceed to infect people in the west and the east sides of the market at the same time. And then 1 week later, another employee that now they're working with lineage A in the lab, And this lineage be the same thing. Full PPP, no symptoms. Lineage, oh, I need to correct this. Lineage A infected. I'm so happy I'm going over this because now I caught that typo. Lineage A infected, never seeks medical care, no antibodies corrected, and it's the same thing, right? They have to do the exact same thing. But the point here is that they have to somehow, with all of these things going on, also go and infect all these different places at the same time. Other than, and this is the natural origins theory, what we have evidence for, at the one on seafood market, infected Siberian weasels, raccoon dogs, among others. Infected walking by humans that were buying them or walking by their cages. And this infects the people on the west and east sides of the market. So, one is extremely elaborate and unlikely, I would argue. And the other one is a lot easier to understand. Even if that doctor did catch it, did he catch it there too? Which doctor? Oh, the people at the east and west sides of the market? Yeah, they would have had to, they'd catch it there. Or are you talking about the Institute of Virology worker? The Institute of Virology worker, if it's the lab leak conspiracy, then yeah, well, if it's at the lab, if it's at the lab leaks conspiracy, they are the carrier, right? They are carrying it with them. And the natural origins, there really is, you don't have to have a doctor or anything working on it, which is kind of the point. It's actually most likely, and this is in the presentation that I'm going to give, or in the lecture slides, the majority of these, so basically over, about 2,000 animals over, all of these different locations, they actually like surveyed and where a lot of these animals come from in these wet markets. Majority of them are actually bred. I didn't know this until this publication, which is I thought that they were just kind of like wild caught or something and brought in, but it's actually overwhelming. They're bred. And this actually coincides with a lot of what we see in the United States with the meat market. Not the meat markets, the meat breeding centers, if you will, which is what they're called in China. So breeding centers are just, places where you're just breeding a lot of the same animals and you get outbreaks of a lot of different types of viruses. That's why the United States uses and pumps these animals full a shit ton of vaccines in order to prevent this from happening. But still, it does happen. So, yeah, the big thing to take away is that there doesn't technically need to be anyone that is taking it from the lab to the other area. But these people, now they're going to be infected and they're going to go out, and the evidence shows that it's the epicenter for the outbreak was the one in seafood market. So we know that that was the epicenter for the outbreak itself. So yeah, so I'm going to make this. Here we go. So before it was text-based. I had written, well, actually it was Marcus that had written it out. Dr. Boom, we're actually going to go over at the end, we're going to go through falsificationism with Karl Popper. I'm going to show and why there is a actual story with actual data to go through this hypothesis. If you're not actually supplying anything with a testable hypothesis, then you can be discredited. And yeah, so that's the kind of the cool part of it. Yeah, bringing it back. Where is this? I think. Let's make the best case scenario. Yeah, where do I put it? Probably here. Maybe at the end. Here we go. There we go. This. Okay. I mean, it really is so crazy to think that one has gained so much traction despite all the evidence to the contrary. It's unbelievable to me. I understand that it's like easier to really grasp the lab leak, because it's just isn't it a coincidence that? But again, through falsificationism, are you actually providing a testable hypothesis? Just saying, oh, isn't it a coincidence that is not it's just not science? So we can't. It's not okay. And these types of science denialism is spreading into vaccines. It's spreading. The amount of Republicans that are vaccine hesitant now, or vaccine skeptical, that don't believe in vaccine requirements in schools has doubled in the last 3 years because of this. So this isn't just an innocuous kind of thing where we're just like, haha, isn't this funny? It's like we're actually having huge implications already, because of this. And it's just not okay. It's just not okay. So we have to fight back. We have to say stuff. This is worth fighting for. So cool. Well, there you go. That's cool. Why are they different? The one, oh man, you could just tweak with figures forever with just for. Oh, I'm noticing all these things that are like weird. That's so funny. Whatever. Okay. And I need to really I need to reduce the amount of slides here. So this is kind of like the rough draft of the, you get like a rough draft of the presentation, and then go through and just call things that I think that aren't that interesting, or there's a lot of papers that I'm going through here, so I have to make a specific point from them, or call them, or call the slides. Yeah, it is weird times, man. I think it's it's honestly really weird to be, as a scientist, getting involved with science communication in such a weird time. I had, obviously seen and created my YouTube channel because I saw a huge event going on, like a huge pandemic. And I thought we would be more readily accepting of technology in ways to actually help us. Like I actually have a video. We're talking like months. I mean, I'm talking about like May or something, or I don't even know when. It was my old channel name and all that kind of stuff. I think I might have privated the video, but I'm reading these news reports of like MIT developing new apps in order to track your spread, so you can be notified of where, what's happening. And it's like, oh my gosh, like that'd be such a cool way to prevent super spreader events and that kind of thing. And I thought people would be more accepting of the technology, but as people grow skeptical of surveillance and GPS locations over time and history, um, we just never accepted and adopted that for the pandemic, which blows my mind because it's like Apple and Google already do that with your phones. That's what I don't understand. So we can have that technology being used for corporations so that they can make money, but we can't use it for public health. Like I don't get that disconnect. Dr. Buenbuen, how did you conclude that the infectious person would have to spread on both east and west sides of the market simultaneously? That is a great question. So basically, this is Warby and Picard. So Picard was what we were looking at before. So Picard states that there's two distinct genetic lineages that happened at the same time, which is kind of crazy, right? Because in one hand, we're like, this is a super rare event that happened. There's a spillover event, and we're saying, oh, wow, there's not one, but there's actually two that are happening at the same time. But since we know that evolution happens within populations, right? Within viral populations and just general evolution happens within populations. There actually isn't that crazy that there would be a couple of them that would be able to make that jump. As soon as the mutations accrue inside the viral populations that allow for that outbreak event to happen, it's just human contact that is the limiting factor. And we had the case with two lineages, right? So we have that. So we know that lineage B went first, and we know that lineage A went second, and therefore we can track those strains and we can track the cases over time. So what we can do is we can actually do environmental sampling of these raccoon dogs. So when I was putting the raccoon dogs in cages, I wasn't joking. We actually have pictures of them. This is before they took it away, though. So they had positive samples here and over on the right hand side of these environmental positives that happen. So like this one right here, but we know that there are specifically a lot of them were in this corner. So the red heat map. I didn't know that these were duplicated. Cool, we can just remove that. And it gets worse. So we can actually have risk and density of the populations over time. So we can actually, the environmental samples can you can look at it over time. Human cases on or before December 20th or after December 20th. And you can actually see that there's on the west side and the east side, there are positive cases. And again, this is another density heat map, basically showing that the west side had it on, looks like before December 20th or an after December 20th. So there was definitely like the basically the current hypothesis is that the outbreak happened the last week of November, basically. And then lineage A came about a week later. So those two things coming together. And where did it go? Yeah, this is too busy. I need to like really. And then here, this is super crazy. So this is really cool. So basically, okay, so what happened? How do we get this data? So after this happened, obviously this is an outbreak event, right? This is insane. So China is like, let's get rid of everything. This is an outbreak that's happening. And so they got rid of these raccoon dogs and all the specimens and stuff and cleared it out. I mean, and this is where kind of the in good faith like questioning comes in of like, why would you like get rid of all of these samples and stuff? And I can see China's case in point being like, we're not going to just let all these and outbreaks continue to happen just so you can do your environmental sampling. But what we do know is that basically after the outbreak had happened and all the animals when it got cleaned out, we did sampling, or scientists were let in from the WHO to do sampling of the cages, of the trash can, which is, we got some SARS-CoV-2 positive there, and of all these different little places in the market. They later took some of that sampling. They actually did sequencing of it. So they did spatial distribution. They looked at the mitochondrial DNA. So SARS-CoV-2 is a RNA virus, right? And really interestingly, they actually did mitochondrial DNA sequencing of the samples. So in these cages, there are mitochondrial DNA samples of raccoon dogs and SARS-CoV-2 positive samples. I mean, you know what I mean? Like how else would you explain that other than natural origins? That's why it's like literally as ridiculous as like this person needs to come in, and that's why it's like literally spitting and coughing on these raccoon dogs and these weasels specifically, and the cages, because that's the only way that you could actually get mitochondrial DNA on the same place that you would actually get SARS-CoV-2 virus. It's pretty wild. So we have SARS-CoV-2 sampling, SARS-CoV-2. I think this is qPCR data. I'm pretty sure, I'm not 100% sure. I need to check that. I think it's qPCR data, but then they did sequencing to show mitochondrial DNA sequencing of raccoon dogs, of hedgehogs, of porcupines. And then they looked on the west and the east side, and they've actually found mitochondrial DNA and RNA virus sampling at the same time. So it's pretty nuts. Pretty nuts. Yeah, it's pretty cool. It is pretty cool. I don't know why the chat's not showing up. So, and this is a great example. This paper right here is a great example of how science is inherently political, right? It is political to take those samples, right? It's political to analyze those samples. It's political to release the data from the samples, and we have to be cognizant of that, right? We have to be we see that, and we see that because the paper that this is based on, the data comes from a preprint server that the Chinese scientists actually uploaded, and then they removed the data trying to kind of like I don't know what maybe it was too, I don't know. I'm just speculating at that point, but they basically took they took it down, and it is a huge thing right now. So that paper actually isn't capable of being published right now. So it's pretty nuts. So that is why science is inherently political, right? We, as scientists, we're not like above the fray or whatever people try to say. It's the opposite, really. Yeah, I think I understand what you mean. You know, I think it's one of those things though where let's see. I think it's just inherently gonna happen. You know, it's just the reality of it. I think yeah, I don't know if it is possible for science to be completely apolitical. You know, it's I mean, think about the conditions that existed you know, before the scientific revolution, right? Where you had theocratic governments, you know, and even, you know, once the scientific revolution started, you know, the idea that there are alternative ways of the earth to form in relationship to the moon and all that kind of stuff challenges, you know, notions of that oligarchical power structure, be it a theocracy, be it a dictator. uh you know, it's there's going to be powers that don't want that data to come out. So I think that science inherently almost has to happen in a libertarian or liberal, a small L liberal society. I don't know if it can exist in anything else where you know, free speech is really fundamental for science, I think. So okay, so let's go through here. Okay, all right. So we're gonna start. I'm gonna give a rough rundown of this. The science. I don't know what to do about my background and my theme. I hate it. I hate my theme. But this is distracting from our needs, and we need to use this to cultivate the kind of literacy that allows free speech to function appropriately. Yeah, I agree, you know. Um I think that this outbreak, just like MERS and SARS-CoV-1, you know, this is going to happen more often in the future, especially with, you know, new novel viruses unfreezing in the permafrost and all that kind of stuff happening, and animal populations are being pushed into human neighbor places where humans are living because we're destroying their habitat. So that's, you know, that's actually the first part of the presentation is basically showing that, you know, basically these horseshoe bats are kind of where people think, you know, a lot of this stuff is coming from, and for good reason too, because these horseshoe bats are live in caves. They have lots of which is, you know, really a good environment for a respiratory virus to live, right? Where there's no airflow and that kind of stuff. So this is only going to keep happening more and more. I don't, you know, and it's kind of a bad omen. I feel like, you know, we figured out MERS and SARS-CoV-1 pretty well. You know, it wasn't so uh, conspiratorial. I don't understand where all these conspiracies came from just because of, you know, these of this virus. But it is what it is. I hate it, but it is what it is. I mean, I think it's. I think things aren't gonna die down anytime soon. We kind of it's, you know, I think it's, you know, the difference between SARS-CoV-1 and MERS isn't like science being more political. I think it's all has a lot to do with social media. I think it has a lot of people that have like not the virus but the thinking. Yeah. the thinking behind it. Yeah. Oh, okay. I understand what you mean now. Okay. Yeah. So, um, yeah, I think, I don't know, man. I think like there's a growing gap that's happening, with science and what people are kind of, they know about, and, it's only getting worse and worse and worse. The, it's honestly unbelievable. I don't even, it's science fiction to, even think about a lot of the stuff that goes through. Um, yeah, exactly. We are leaving a lot of people behind. Exactly. And I think that that's kind of the big problem that we're having, is that now we've not taught molecular biology, we've not taught genetics, we've not taught, RNA, or nanotechnology to people, and now, we're bringing in RNA nanotechnology delivered biomolecules, and people are freaking out. And, meanwhile, I mean, this nanotechnology research, the lipid nanoparticle delivery that they've used in the vaccines, is old news. It's 30 years old. We're doing way more cool stuff in nanotechnology now than, than this. But, people freak out. They are, it's pretty nuts. Change theme. I wonder if I could do something. I don't know if I like the black backgrounds. I usually, in my presentations, I'll do black backgrounds, but for monitors, I actually like the white. It's like more clean, and it gives higher contrast. And my figures have a white background. Okay, so let's go through so I'm going to go through a whole spiel on this thing, see how long it is, and kind of like take notes and see where I can. I don't know if I'm going to do it tonight. We'll see. The video tonight. We'll see. Okay. All right. So the science of SARS-CoV-2 is a lot different than what the media has portrayed and what we kind of talk about. But I think it's really important to show that the science of actually, what we know and how we know what we know. Um, so in general, let's see. So in general, where do pathogenic viruses come from? So basically, there is a cycle that happens between viruses that live within these long-term host reservoirs. So, for example, this could be, a bat. This could be a different type of mammalian reservoir, like a rodent. And within these populations, these viruses evolve. So they evolve different coat proteins, different ways of infecting different cells. They will infect and go from mammal to mammal. Then they will create new mutations in their genomes and accrue these mutations, in increasing their genetic diversity. So genetic diversity is very important for viruses because it allows them to, if they can't find the host of, their preferred host, it allows them some wiggle room. So now that they can find, maybe if they can't find their preferred host, they can latch on to another cell and another reservoir, so that they can continue mutating and spreading. So within these reservoirs, they have infectious parts of them, of their life cycle, that aren't necessarily like, they're not going to kill these bats. They don't have necessarily are going to be have symptoms, for example, but they could have be accruing mutations that still latch on to their virus, their cells. And these mutations and this genetic diversity eventually will actually create a novel variant that has these transmission potential. And transmission potential is the ability for them to actually go into a new population, like we were talking about. And So you could have a bat, for example, that has a coronavirus, for example, that then goes into another mammal, for example, like a civet. So in this example, you could have multiple multiple bat coronaviruses infecting some type of like civet or a ferret, and then that would allow them to recombine within that organism. And then that recombined virus would then spill over and create a new novel virus that could infect humans. And this is kind of like what we find. So the history of coronaviruses that have been linked to outbreaks kind of starts with MERS. So MERS is where a camel was shown to be a reservoir. This was in 2012, and Saudi Arabia in 2018, with less than 1,000 deaths associated with it. And then there was SARS-CoV-1. So then SARS-CoV-1 was obviously impacted humans, but this actually had the long-term reservoir of bats, and then finally went over into palm civets, which is like kind of like a ferret. There's a picture here. And this is known as the 2002-2004 SARS outbreak. And this led to 700 deaths and about 8,000 confirmed cases. Now, like more generally, coronaviruses actually infect a lot of different types of organisms. It's actually kind of interesting that they are able to infect so many different ones. So, for example, Delta coronaviruses infect bird populations. We have gamma coronaviruses that will actually infect mammals and marine mammals and birds. We have alpha coronaviruses, and then there's also beta coronaviruses. And beta coronaviruses are what we actually deal with SARS-CoV-1, MERS, and SARS-CoV-2. And if we actually sequence the genome of these different coronaviruses, this is what we get. So SARS coronaviruses are actually pretty interesting genetically, because a lot of their genome actually encodes for proteins. There's not that much space that is called untranslated regions, or UTRs here. So you can actually line up these open reading frames. Open reading frames lead to proteins, and these different parts of their genome can confer these proteins that we see. So, for example, this S spike protein, this is actually what gives the coronavirus its name. These little spikes that give it its crown, and this is the part of the genome where it comes from, right? So this is the spike. And there are different proteins that are made within here. And this is what we do. And this is in environmental microbiology. This is what we're actually sequencing when we are sequencing these viruses. And we can take these regions then of these viruses and we can align them. So using computers, you can through next generation sequencing, we're taking millions of pieces of RNA that will align nucleotide by nucleotide, match them up. And we can actually see how these regions will evolve and what they come from. So this is where the SARS-CoV-2 genome comes from. So, this first region right here has this human SARS-CoV-2 sequence, and then this rat G13. So this is rat G13 is interesting because it's a collection of RNA sequences in a bat that we found, but it's not a virus necessarily. So it's from bat origin, but it is the most closely related genetically to SARS-CoV-2 that we've found in the environment. The rat G13 is about 40 years distinct from SARS-CoV-2 in the environment. So region 1 can align with pangolins. And think about when we're looking at these different regions, think about the amount of recombinations and the amount of different ways that these genomes can interact, because where all these different parts are coming from, they're different, right? So we have this second region that is from related to bats and pangolins. Then we have this third region, which includes the spike, which is very important. That's the part that actually latches onto cells. That has a SARS-CoV-2 in pangolin and rat G13. But interestingly, this is actually more related to pangolin than rat G13, which is interesting. And then again, the human SARS-CoV-2 with rat G13 in region 4 with the nucleocapsid. So what can we learn about this long-term host pathogen coevolution area? And what would we need to do in order to figure out what's actually happening here? Well, we would actually need to do sequencing of that. So we would basically need to be sequencing these bat and rodent populations, so that we can kind of figure out how these genetic mutations are happening and how they're accruing. So this is a really interesting paper that actually looked at coronavirus, if we could actually find SARS-CoV-2 or related coronaviruses in bats in Cambodia samples from 2010. So this is where Cambodia is on the map in relation to into China. And interestingly, they didn't find SARS-CoV-2, but they found a very closely related genome here. So this is right here. This orange right here. This is these, and this is a phylogenetic map. So phylogenetic maps mean that when you're close to one another, you have a lot more significance of being related to one another. So this is SARS-CoV-1 up here, for example, and then a lot of genetic diversity with all these different branches and lengths here. And then we found that there is a very, very similar coronavirus in Cambodia in 2010. So how does this genome actually look like? What does it look like when we actually sequence it? And this is one of the reports that we would get. So this is basically looking at the similarity nucleotide by nucleotide and putting them one on top of the other, trying to see how divergent each of these genomes are. So as you can see, they're not all the same, right? These in any type of population, you're going to have genetic diversity, and genetic diversity is good for any type of organism. And what we see here is a lot of genetic diversity that's happening. And then right here, this dip right here is a dip at the spike protein. So again, spike proteins needs to be very specific for SARS-CoV-2, for the ACE2 inhibitor, the ACE2 binding domain for humans. And this is what we saw. So what I'm seeing here is a lot of genetic diversity, right? These sequences are similar here, and then you see the rat G13 not dipping when everything else dips, because, well, rat G13 is the most similar to SARS-CoV-2, so that kind of makes sense. But what we're seeing here is a lot of potential in the ability to recombine. It's kind of like a lot of genetic components that can be used and configured in any which way to create SARS-CoV-2. And that's what we're kind of seeing here. And each of these parts of the genome can actually be looked at piecemeal. So we can actually look to see actually where, how similar each of these parts of the genome is to SARS-CoV-2. So on the bottom here, you have SARS-CoV-2, rat G13, and then these new viruses that were found in Cambodia in 2010. Again, they're very, very similar. Each of these, this one not so much because it's a farther distance. But the idea is that even back in 2010, there was the genetic material needed in order to recombine, in order to make like SARS-CoV-2. So we've seen and we can look back posthumously, right, to kind of see where we can detect SARS-CoV-2, and that's basically what we're finding. The amount of building blocks, the genetic building blocks that exist within nature, is there. And let's see. The spike looks like it had a hotspot of activity for a while. Can we analyze for that soon and implement more localized measures before a pandemic hits? Yeah, so that's like a really interesting question in terms of RATG13, right? So RATG13, basically one of the biggest questions that we have right now is how can RATG13 actually infect humans? And that is something that we don't know. We would basically need to take the RATG13 sequences and kind of like put it into the it's a different virus, but one that's been genetically modified that we can actually change and then change little bits over time to see if something like RATG13 could actually be infecting humans. So we would, and that is the gain of function things that it's incorrectly named that, but that is the research that people are trying to stop from happening, unfortunately. So that is kind of the political problem that we're having now, is that the research that needs to be done in order to see that what mutations in the spike would actually be causing an infection into humans is not being studied because of politics, which is ridiculous. A good question. So it's interesting that we can actually look for, so we have bats now that are, with these coronaviruses. We know that SARS-CoV-1 had bats in it. So how prevalent, like, are bats and coronaviruses? What can we tell from the sequencing of bat populations about coronavirus genetic diversity here? If we're looking at Cambodia in 2010, and we have lots of genetic building blocks, like how prevalent is this across all bats? So this is, this study took a ton of different samples from a lot of different bat caves, did some incredible work that was, I believe, right during the outbreak that was happening. And basically they mapped on the coronavirus genomes that they found. They aligned them, did this phylogenetic analysis on the top right, and then actually mapped onto the hot spots for a map in China to actually see the geographic range of these bats. Because bats, they are, and as we can see here, it's pretty nuts. There's up to 23 different species all in one space, geographically. It's across. So these populations of bats are linked together, not like, from one side of the map to the other. Bats, they have shorter wings. They're not doing these crazy migrations. They'll, go from one cave to another, but they're, pretty, they do have, some genetic wiggle room. There's immigration and emigration into each of these bat populations, as we can see. So, but, overall, they, through the genetic richness of having all these different types of different species all in one place, that basically allows an accumulation of mutations for that recombination event to happen. And again, we can look at these sequences now and we can see basically the differences. So what one would be SARS-CoV-2, all the way across, right? We see RATG13 again, having the most similarity in the spike. But I think the most interesting part here is that they're not aligning up, right? We like the other genetic map that we just looked at. This one is actually showing more genetic diversity, right? Because these these populations. They're a larger amount of populations that we've seen previously. So again, this is showing us, and we can actually look at it just like we had seen before with that phylogenetic map. We can actually, this is like a kind of a data table with the the red indicating more likeness and blue showing less amount of genetic alignment. So what's the most interesting to me here isn't necessarily that they didn't find rat G13, they didn't find SARS-CoV-2, right? But the interesting part here is, again, it's just more evidence of all these genetic building blocks that are capable, and the genetic diversity of these bats. They're not secluded into just one cave and where you see the same coronavirus across all of them, right? They are introducing these viral populations and this genetic diversity at an astonishing rate. It's pretty crazy. And this is allowing them, and this is one of the reasons why you see coronaviruses in so many different populations, is because of this, right? Because of these genetic diversity. This genetic diversity is what allows them to go from one spot to the other, and one host to the other so incredibly well. It is this ability, this genetic diversity. Okay, so what can we tell now? Okay, so those are the bats, right? That's the background information, right? That's what's going into this intermediary organism. So the intermediary organism doesn't have to be a bat. In SARS-CoV-1, it was a civet. And basically these mammals are getting introduced to bats. Bats caves are being destroyed because of humans wanting to live there, for example. And these are kind of like the intermediary where finally humans actually interact with this organism, and that's where the outbreak happens. So what can we tell? We've looked at the bats. We've looked at the first couple stages here. What can we talk about the step more closely related to humans, and finally how we actually get the pathogen? Well, that's what this amazing paper is. Virome characterization of game animals in China reveals a spectrum of emerging pathogens. So in China, there is a kind of like a web of how these people interact with mammals or animals more generally, I guess. So we have humans at the bottom with domesticated animals, and these animals, domesticated animals and humans, actually they deal with the trading of game animals through wet markets. They also breed these game animals in breeding centers, like farming. And then also these breeding and game animals and these trade game animals are introduced to wildlife animals, right? Because they're not in sterile places, right? They're breeding them and trading these game animals, wild animals, from animals that are interacting with the world. And these animals that are interacting with the world will come by and cough or breathe on these breeding centers or interact with other trade game animals. And then that will actually kind of intermingle with these wild game animals that just exist in the wild. So there is kind of like this complex web of viruses and virus outbreaks that happen inherently because of this. So there's basically tons of recombination and genetic mutation events that can happen, right? You're getting new genetic material from brand new coronaviruses. There could be crossover events that happen that you get introduced to even more viruses. So the point is that there is a lot of ability now to get infected with this new genetic information. So this is the paper. Over 1,900 animals. They found 102 different viruses and 65 viruses for the first time. So these are the enormous amount of work over all this, this area right here, all the different types of species. And this is one that I wanted to highlight here, larvata, excuse me. That's the ferrets that we were talking about. That is SARS-CoV-1, right? That's our background. That's what we know going into this, is that, well, SARS-CoV-1 was in a bat. Then it was in what's the name? A civet, larvata. So it's a civet. So this civet we know that is an intermediary that caused SARS-CoV-1. So let's take a look at it and watch it. And in these wet markets, remember, they're going there and they are buying them and they are watching them for symptoms to see if they're healthy or unhealthy. And then they're also looking at the living conditions that they were found in, that they came from. So something that I just learned from looking at this and reading this paper, a vast majority of these animals in these wet markets are actually from breeding centers, right? They actually breed these for food. This isn't just like them catching something. This is them breeding these specifically. So this, again, high density of animals. So it's easy for a respiratory virus to go from one animal to another. And then they bring these infected animals into the wet markets to sell. So, and this is crazy to me. I'm never, very few figures have actually taken my breath away, like I did with this when I first saw this. So this is a crossover event. They actually found, with this paper, they actually found one of the crossover events. This is inside of a civet, which is the intermediary that was for SARS-CoV-1. Remember, SARS-CoV-1 was in a bat reservoir. The intermediary was a civet, then into humans. They actually found a crossover event of a bat coronavirus inside of a civet. So this is, this could be SARS-CoV-3 or 4 or whatever. This could be part of the sequence, just like Ratchy 13 40 years ago. This could be the start of yet another crossover event. So this is a, basically this larvata, this civet, if they had the genetic capabilities of interacting with a human cell, I mean, this is exactly how this stuff happens. They literally found it. They saw it. Just an unbelievable finding in this paper. It just shows how scary this stuff is. It shows how insanely prevalent, right? It's just unbelievable. The civets are one of the highest trafficked, game animals, so it's not super unsurprising. But look at all these unhealthy animals. Look at these unhealthy animals that are there. It doesn't take a lot. It doesn't take a lot of them for in order for this to happen. So, what do we need? What do we need to do? This is just a small sample of animals, right? This is only 1,900 animals. They had a great geographic distribution here, but the problem that we're having is basically the amount of genetic mutations because of coronaviruses and how they are capable of going from host to host to host. we have a problem in that their genomes are so diverse that it's hard to sequence them. It's hard to sequence all of their genomes at one time. And this kind of goes into an analogy that I have. But first, this is animals cultivated for diversity the same way that most U.S. poultry has salmonella outbreaks. Yes, yeah, exactly. Yep, exactly. Just with an extra step from one. How do we describe it? A second-degree zoonotic source in a profit-cultivated mass spreading event? Yeah, I mean, I think that's correct. Yeah, yeah, it's hmm. I think that there's basically a lot of vaccines and antibiotics and a lot more sterile conditions that happen in factory farming in the US than this. One big difference is that, you know, unfortunately, the pigs, for example, in whatever pig farm, they're not actually interacting with wild animals because they're not even out there, right? They're not even out um in nature at all. So in a weird way, that kind of helps because it kind of decreases the amount of genetic diversity of the viruses, so they can't hop as much. So, and on top of that, they're pumped full of flu shots, and other vaccines and antibiotics to stop secondary infections. So, the problem is that this is kind of the worst of both worlds kind of thing, where you have no medical treatment for the animals, and then on top of that, there's nothing stopping these unhealthy animals kind of going to the wet markets, which is just nuts to think about, but it's true. I mean, it is to a good point that like China realizes that this is happening and is trying to tamper down on a lot of these illegal wet markets that are happening. But I mean, I'm pretty sure that just the illegal wet markets, I heard the stat, just the illegal wet markets alone from China are as big as the factory farming industry in the entire United States. So the illegal wet markets are gigantic. They're as big as all of the factory farms in the United States. It's pretty nuts. So how do we see if these crossover events are happening? How do we see if these events that are causing these these potentially harmful viruses, how do we see if they're actually happening? Well, it's kind of like sequencing. Sequencing, it can kind of be seen as taking scoops out of a jelly bean jar. The ability for you to find every flavor inside of a massive jar of jelly beans is kind of contingent, if you think about it, on how many types of jelly beans there are, right? If there's a giant bowl of jelly beans and there's only one flavor, so one type of bean, how many scoops would you need to get until you got your all of the flavors? Well, only one, right? How big of a scoop would you need in order to actually get all the flavors? Well, if there's only one flavor, you need the smallest amount, right? Just this one little small scoop. Okay, so now let's think about it with increasing the amount of flavors. If we have hundreds of different types of jelly beans in this jar, if I'm increasing the amount of flavors, I'm going to have to either increase the amount of scoops and keep my scoop size the same, or my scoops are going to have to get gigantic, right? And I'm going to have to just take larger chunks out of all of these jelly beans, a jelly bean jar. And unfortunately, we just can't, as scientists, we can't just scoop the entire jar, right? we can't just sequence every single coronavirus out there. We can't just sample every single bat. And just like the amount of flavors, well, the coronaviruses have a lot of different genetic diversity. So it's not just 1, 5, 20, it's thousands of different types of flavors. You know why? Well, it's because of the recombination events that we were talking about, all the different types of interactions that they have with all these different other organisms are creating genetic diversity within their genomes because of recombination events and the accumulation of these genetic mutations. And the amount of jelly beans, how big this jar is a function of kind of like how many bats there are, right? And if there's a lot of different types of bats, like as we saw with all those the geographic distribution, lots of different types of bats, lots of bats, and a huge large amount of coronaviruses. So basically, we're just skimming the surface right now, and that's why it's so important to have environmental sequencing done regularly, so that you can actually take scoops out of that jelly bean jar over time, and you can kind of get a better understanding of what is actually in that jar. So, did we find SARS-CoV-2 in the environmental sequencing that's led up to this? No. Does that mean that SARS-CoV-2 is the precursor? Could be out there? Like absolutely, very easily. Well, why? Well, it's because of the genetic diversity that exists. There's lots and lots and lots of different viruses, the coronaviruses in here that could have the precursor to SARS-CoV-2. And just because we've only found rat G13 doesn't mean that SARS-CoV-2's precursor can't be out there as well. So what do we do have? This is one of my favorite papers that I've ever read on this. What do we do have, though? We have the SARS-CoV-2 genome. So can we use that to figure out something? Can we use the genome itself to kind of let us know more about the SARS-CoV-2 history? And yeah, we can. It's very cool. So it goes into this portion of it where how and why this genetic diversity can increase. Well, it's a function of basically host-specific mutations that accrue through SARS-CoV-2. So this is Sean et al. 2021. Host-specific asymmetric accumulation of mutation types reveals that the origin of SARS-CoV-2 is consistent with a natural process. So RNA-dependent RNA polymerase. So what is that? So basically, how do RNA viruses replicate? Well, they have their own machinery, and they basically create RNA from RNA through this thing called RDRP, or RNA-dependent RNA polymerase. This is a little different than kind of like what you normally see in a biology class. It would, you basically have DNA to RNA to proteins, right? Well, this is RNA to RNA. And basically the problem that a lot of RNA viruses have is that basically when they make their RNA, it's kind of transient. The differences and the mutations that happen can kind of like go from transcription to translation, but then there's no feedback mechanism to kind of like make to accrue mutations in that RNA viral genome. And they want those, right? They want more virus mutations to accrue over time. So what basically winds up happening is that the environment can cause mutations in a RNA virus, and it's actually more so than the RNA polymerase. So, which is kind of interesting to think about, like, what's the environment for an RNA virus? Well, the environment is the host, right? So the hosts can have different types of mutations that have based off of a lot of different factors. So oxygenation, reactive oxygen species, different types of enzymes and all that kind of stuff. Basically, just like how your DNA is damaged from the sun through UV damage, basically this RNA viral genome can change. Actually, the nucleotide sequence can change based off of the environment itself. So these are basically different types of mutagenesis that can happen. That's replication dependent, right? That's from the RDRP. So the RNA dependent RNA polymerase isn't perfect. It's going to have its own kind of mix-ups over time. So it can actually cause a nucleotides change there. But our replication independent mutagenesis is actually where a lot of this happens. So basically these de novo or new mutations happen not as a function of transcription, but they actually get added on and created as the viral genome is replicated inside of the organism itself. So therefore, basically the majority of mutations in this positive single-stranded RNA virus, which is the SARS-CoV2, for example, comes from the environment or the host and not RDRP. So therefore, the really cool part, is that hosts or the environment or the hosts that these coronaviruses are in are actually going to have their own specific mutation rate that we can back calculate to. So what does that mean then? Well, it means that we can actually take a RNA virus and we can take their genome and we can compare it to other RNA virus genomes, and we can see where their environment was. They can see where their host was. We can see where they've been spending their time, and that's what we'll see. Chat. So RNA viruses have built-in mechanisms on how they reproduce that makes mutations inevitable. Yeah. So RNA viruses kind of realize, well, they've evolved to bring their own machinery to make themselves. So that's one of the things that they can kind of they can hijack the nucleotides that are normally made for mRNA and other RNAs in the cell, and they basically take those resources and they bring their own machinery. It's kind of like they bring their own hammer and they steal the cell's nails, and they just kind of hammer in their own genomes. Yeah, exactly. So yeah, viruses are very well evolved in order to take advantage of the host's mechanisms and the host's nutrition or capabilities or their resources. So can we do this with other viruses? So SARS-CoV-1, okay, let's see where it matches, right? We have the SARS-CoV-1 genome, and if it's host specific, then we can actually look at the SARS-CoV-1 genome that's isolated from humans and compare it against other SARS-CoV-1 lineage. And we can kind of look at the mutation rate over time to actually see where it actually stayed. So at the bottom here, we have bats. Okay, that lines up to where we thought, right? It aligns, that mutation rate aligns up with bats. It aligns up with another bat. And then the mutation rate has a drastic change into a civet specific mutation rate. And what's most closely related to the human? The civet, not the bat, because there was some time that was spent inside the civet. So there would be some type of mutation rate difference between the civet and the bat, and that's what we see. So cool. This paper is unbelievable. So let's try MERS. Let's try MERS-CoV. So MERS matches with hedgehogs, and then again bats. Bats are going to show up a lot in respiratory viruses, right? Their lifestyle is just perfect for respiratory virus spread. Okay, so and then now we have a different mutation rate that comes from a camel. And what's the most likely? What's the one that's most related to the camel? The humans. So, and this ties in perfectly with what we know the life process of the SARS-CoV-1 was. Is that it spent time in a bat and then got as the reservoir, long-term reservoir, and then the intermediary was a camel and into humans, and that's what we know. So, what does SARS-CoV-2 line up with? How do we know? Well, we have a bat again. We have that pangolin region. Remember, we were looking at the sequencing before, and we saw some regions of the SARS-CoV-2 genome that aligned with pangolins. So that makes sense. We have more bats. We have that RATG13 again. So it's lining up with the ones that we know are most likely related to SARS-CoV-2. And then we have the ones with humans. So what does this tell us? Well, it shows us that there's basically clumps. These bat exclusive viruses can clump together. These human and camel exclusive ones, clump together. So we can pretty much ascertain now, we can back calculate to see that SARS-CoV-2 lines up, the mutation rate lines up with RATG13. And RATG13 isn't a virus necessarily, it's a collection of RNA sequences that we found in bat populations. So what's that tell us? Well, it means that the long-term reservoir for SARS-CoV-2 through molecular forensics, through molecular biology like we know here, it's a bat. So it just like RATG13, it has those telltale signs of bat mutation rates, which only exists for those hosts. So cool. Such a cool thing. So what do we need in order to study these kind of things? I'm going to put this to the end. We're going to save this one. Okay. So, all right. So what's the story? So what can we take away from this? We just went through the last 20 years of sequencing and molecular biology research on coronaviruses. What do we know? What do we know going into the outbreak, right? So we have a huge body of evidence to show that coronaviruses, beta coronaviruses, and all types of sorboviruses, which is what the coronavirus is, but it's a beta coronavirus specifically. It's infecting a lot of different types of organisms, isn't it? And it's making lots of different jumps, and it's made these jumps before into humans. We know that they have a long-term reservoir in bats that has been shown and proven before. It has intermediaries because of this genetic diversity that exists within these coronaviruses. It allows them to have lots of wide variety of hosts, and that's how they can actually this wide variety of hosts allows it to adapt to new hosts, which is these outbreak events that happen. And we know that there's lots of different types of animals that exist in these regions that can actually active these reservoirs, right? That can act. These mammals are creating these intermediaries that allow this insane amounts of recombination and genetic diversity that's already found in bats to have a new place that has more potential to segue into humans. And we've seen that with SARS-CoV-1. We've seen that with MERS. And these animals that we know through studying bats, they're being driven into human populations. And basically, it's allowing these this ability for these mutations and recombinations to actually latch onto humans. And we know through sequencing that RATG13 is still the closest relative related to SARS-CoV-2, and we found it about 40 years ago. So now let's transition into the outbreak, the actual outbreak event itself. So basically, the spatial and geographic spread of the epicenter of SARS-CoV-2 has been found to be the Huanan seafood wholesale market in Wuhan, China. And how do we know this? Well, we can actually look at the spatial patterns of the cases that have linked to the Huanan market and that have no known link to the Huanan market. And we can kind of tell that basically they all radiate outward from this one spot. They have let's get rid of this. They can use Weibo data. So with a combination of Weibo data and cases that we know that are linked and aren't linked to the market, we can actually create a bullseye, kind of, with this with all these cases, and they all center on this Huanan market. Interestingly enough, so even the ones that aren't linked directly to the Huanan markets, i.e., I wasn't there. They're still radiating outward from this Huanan market, based off of this data that's available. And then we can also let's see. I don't think this is. I'm still going through these, trying to figure out which ones I want to use. Is that why we're expecting a hotspot this summer? Nighttime infection hotspots during the regular summer bat migrations in the southeast. I don't know. That's a good question. I think, I don't know. I don't know about that. I haven't. I don't know. I'm not saying that it's not exist there, that it's not going to happen, but I don't know. Have anything to speak on for that one. I think, I don't know if SARS-CoV-2 has been found in the bat populations in the United States. I hope not. I really hope not, because if they are, then that's really bad. It's a really bad sign. Yeah, that would be really bad. I know that there are reservoirs of other organisms that aren't humans that have SARS-CoV-2 in them, but basically right now, since we're still in pandemic, basically the COVID or SARS-CoV-2, SARS-CoV-2 just has a ton of mutations that are still happening. So you can actually go to next strain and covariance. This is what I used in order to track the kind of like what's actually happening. So you were talking about in the US specifically. So here, let's do this. All right. Boom. Okay, so you were talking about the US specifically. So on the data set on the left-hand side, you can actually choose just North America. So we'll do that. And then we can zoom in here. The Northeast. Wow, that is a pretty high amount of genetic diversity. So this is kind of cool. This is the genome of SARS-CoV-2 itself, and then these are all the different mutations that have been found. So you can tell, like, again, the spike. This is actually where that dip is when we were looking at this. Right here, this dip right here is another way of looking at it. Is this these mutation rates are going to decrease similarity, and what this is a function of is similarity. So in other words, this is an increase of mutations in this region of the spike protein, and that's what we see right here, which is kind of cool, right? And interesting. So I would be looking for like a exponential increase in a new clade in order to look at if we're going to see a huge increase, and I would expect to see that's not to say that it's not happening, right? That there could be a lower viral population that's going up, but I don't see one that's like kind of in that huge influx. Um, but the problem is that we aren't living in a vacuum, right? So it's the entire world, and that's why it's a pandemic, right? A pan is globally. So basically, all of those different types of new strains can be coming through with other parts of the world, and then basically recombine with and spread in other parts of the world. Is there a rough centroid region in the genetic diversity in the US? Yeah, so there's here. So this is sequence frequencies by clade. So this is, you can see here that the clade's 23A XBB15 in the northeast. I think it's pretty much dominant for everything. Is this um is this one? Or I'll link this in the chat so you can see it. I wanted to look at it too. And then this is the covariance too, which is kind of cool. So this is XBB 23A. Here's the Omicron variant that's the one that's the prevalent one. I wanted to do some research on it. 23B is the one that's kind of coming up. That's this Omicron variant. It's Omicron variant because of this right here. This is a phylogenetic map. So these are all differences that are kind of bound to one another. And they're all in the same branch, which is why they're Omicron. They're all Omicron variants right now. Yeah, so this is the same. So that's the information that we have right so far. Not to say that like this, it's going to stay this way, or basically new variants can just take off, which is kind of interesting. Omicron was, if you could look back, you could see that there's like a lot of genetic diversity here. What's going on here? Like, why can't I click on it? Weird. Which other species share the Omicron variant with us? Which other species of organisms are infected by Omicron? I don't know. That's a really good question. It'd basically be to anything that has similar ACE2 to us that we know of, unless the Omicron variants allow it to latch on to other parts of another animal, another cell. Basically, that would be the stipulation. I don't know about that. You basically would look at we can try that. Blast search. What's the ACE2 human peptide sequence? So this is the ACE2. I need the sequence. Oh, actually, I want the amino acid sequence. Not getting it. Down, send to clipboard. protein blast. Oh my gosh, it didn't copy my kit. It lied. Download data sets, gene FASTA, protein sequences FASTA. There we go. Oh my gosh. FNA file. It's usually just text. Don't do things live. That's what I'm learning right now. Gene result, notepad. I'm looking basically for the string of amino acids that's created from the from DNA, and then we can align that with other sequences to see how similar they are, and if we could actually. It's annoying. I guess they're like super divergent then. Wow, that molecular weight is gigantic. Maybe that's why. Let's see. I'm sure that, I mean, this is actually the first part of anything related to this. Let's see. I wanted to try to just do this live, but it's not looking like it. I'm stubborn. I might just keep trying. Oh, looks like someone beat us to it. Dang. Here we go. There we go. We were looking for the ACE2 sequence, and there we go. We found it. So this is the ACE. So this is what I wanted to do. So basically, there's different parts of the ACE2 protein, and this must be the binding site for the glycoprotein of SARS-CoV-2. So this would be other things, other organisms that are very similar to us. So these similarities decrease. So, I mean, basically any of these would work if there was enough genetic variability within the SARS-CoV-2 populations, that I would think that any of these could actually work. Oh, okay, cool. Protein FASTA is probably what we would want. Yeah, that's what I was trying to do. Yeah, I would recommend probably looking at, the paper is probably going to be better than just me doing it ad hoc, even though it's fun for me to do it. But, dang, dude, what a brief research report. This is something that they did in July 2020. Lab is closed for the summer, and they're like, what can we do? And they just did a paper on this. I wish I would have just wish I would have published a paper like this. It's like, no, all downloadable stuff, all downloadable things. So this is the ACE2 protein in humans and how it relates to other things. So yeah, civets. Wow. That we could have just hypothesized that one on ourselves, huh? Cattle, goat, horse, horseshoe bat. Wow. I mean, are we surprised? Are we surprised that horseshoe bats and civets are on here? No, we are not. No, we are not surprised at all. Because that is exactly what we would have expected, right? We would have expected those to have, we would have expected it, and we did expect it, and we saw it. That's crazy, though. But I don't know. I don't know why I'm surprised sometimes. It is important. Peer review is very important, and the problem, Frontiers is legit, and it's one of those things where, like, I can just add the problem with. I can tell you right now the problem with if I was just doing an ACE2 protein This is a really long protein. It's very large, 805 amino acids long, which is very, very large. Not having these binding regions, that would be the biggest one, because if I just aligned it with BLAST, basically these differences on different parts of the protein could have been different, and I wouldn't have known that. So they aligned it and they looked at the similarities specifically with the SARS-CoV-2 glycoprotein region, which is better than what I was going to do anyway. So give him props for sure. Okay. Dang. Okay. Okay. I'm going to do this. Delete this. Okay. So we have cases from WHO data that are linked to Huanan market and the ones that aren't, and Weibo data that, again, puts a lot of this data together. And when we put it all together, then we can see basically a linked cases and unlinked cases, all with the epicenter, kind of like a bullseye, all coming in right onto that that market. Let's see here. Okay. Okay. And well, how prevalent is that? You know, how prevalent is that, you know, people winding up going to that area? Because if all these cases are centered around this Huanan seafood market, you could say, well, it's a really popular destination. A lot of people were going there. Everyone's going there. So of course you could link people like to that. Well, you know, using the same type of data, right? With Weibo and their the type of an ability that they have for GPS tracking. You have all these different locations that we can look at. And there's high-rise locations, there's markets and schools and buildings, hospitals, residencies, and then the Huanan seafood market. So we're talking about a ridiculously small and low-trafficked area inside of China and this place that kind of shows that they were going there specifically with amongst markets and shopping malls. You're specifically going to Wuhan, and it's not a coincidence that it just happens to be there. So another point is, well, okay, so what we know, if Huanan was the epicenter, what kind of samples could you take? Well, after the outbreak happened, they were able to go in, and the WHO was allowed to go in and create and take environmental sampling from these different stalls that had different animals into it, and they found site specific, and amongst different cages, amongst all the different west and east sides of the Huanan seafood market. You had SARS-CoV-2 samples that were found, and you can actually even look at the density in particles over time. You can actually, these are the relative risk and the density of these samples that were found. So you can tell that there was actually a clustered, specifically in the west side of the market. There is actually even a clustered even amongst that versus the east side. And we have an ability to even look at it temporally, because the different strains on or before December 20th, which is, we know that there are two distinct strains that were part of the outbreak. So we know that the first set which is the light blue here, was more on the west side, versus the cases after December 20th went into the east side. And this is the cases over time. So we can see, again, the density of the particles and the positive cases and the outbreak going up over time in the east side. So if we looked at these through sequencing, we know, like I had mentioned before, we actually have two separate strains that actually were part of the outbreak at the same time, which allows us to have more of a fine-tuned ability to look at it over the entire outbreak. So this is another phylogenetic tree. But basically, each of these lines show any type of genetic diversity that would happen. So there's a distinct lineage A and a distinct lineage B that could only be from two different strains or lineages of SARS-CoV-2 at the same time. These are probability distributions that happen over time. So these are the ability for the probability of these phylogenetic structures that we saw here to actually accrue with it, be it from a single one mutation and then another released over time, or two simultaneous ones that were released. But the only way that you actually reconstruct the phylogenetics that exists is to basically do what happened, where you release one and then you release another a week later. And we can actually even see the cases over time. So you can actually take the density and the genome sequences over time, and you can actually see how B comes first. And you can actually create a timing specific event that happens specifically because of these one strain coming first and then the second strain. And this is a mathematical interpretation of it, and that basically this could only arise from the one strain and then the second strain. Finally, there was another paper that came out. Let's see if I have this. Zenodo. Okay. Let's see here. This. All right. So this is data. Is Werribee on here? There he is, Michael Werribee. That's awesome. New data. Overlapping mitochondrial DNA and RNA and SARS-2. So this is the only possible. These are mitochondrial DNA samples of these different types of species, and this would have to be mitochondrial DNA from these organisms that were actually overlapping with RNA from the SARS-CoV-2 virus. And if you do a phylogenetic analysis of these samples of the mitochondrial DNA, basically they only come from they're super highly correlated. We've had tons of media talking about this. Unfortunately, none of them have really talked about any type of actual evidence to go into these lab leak conspiracies or talking about any of the data that I've presented to you just now about anything to do with viral evolution or genetics whatsoever. This is one of the first reports that happened with the Republican House of Representatives in the United States of America, talking about the origins of COVID-19. And this thing is 90 pages of nothing, no actual evidence that actually ever that is ever used or presented, which is incredibly frustrating. And this is the only part where they actually even have any type of genetic evidence that they're actually trying to put. And this isn't even SARS-CoV-2 at all. This is actually just Barrick's idea on how he can stitch together with PCR these different parts of these genomes in order to actually create a viral genome. This isn't even, they're not even using it or comparing any type of actual biological data. There's just emails for the all these other pages that aren't even worth looking at. But this time, and you might have seen it in February 2023, there was another DOE report that was talking about that this was going to be the whole idea of where this thing came from. And yet the Department of Energy has no actual report to link these lab leaks together. They present just no report, no data, no publicly available data that can be peer reviewed. And again, we have another office coming out saying that there is a potential, releasing a report saying the potential links between the Wuhan Institute of Virology and the origin of COVID-19, the pandemic. And this is, again, just having to quietly deal with these, the facts of the matter of the case, and having to conform to reality, where the National Intelligence Council and 4 other IC agencies assessed that the initial human infection with SARS-CoV-2 most likely was caused by natural exposure to an infected animal that carried the SARS-CoV-2 or a close progenitor, a virus that probably would be more than 99% similar to SARS-CoV-2. So already walking back this lab leak, confirmed idea from the Department of Energy. The Department of Energy and the FBI assessed that a laboratory-associated incident was the most likely cause of the first human infection with SARS-CoV-2, although for different reasons. So they can't even confirm within each other their own conspiracies that have zero factual data or scientific evidence to show. And again, the CIA and other agencies remain unable to determine the precise origin of COVID-19 pandemics. Interesting, since I'm sure they're wiretapping everyone. I have no idea why they can't present any of this data. And again, almost all the IC agencies assessed the SARS-CoV-2 was not genetically engineered, which is funny considering how much of the House committees and the Senate committees have been talking about how this was a product of gain of function research. This was a product of all that type of genetic engineering. So they're already having to walk back these statements, and they're having to face reality. Most agencies assess that SARS-CoV-2 was not laboratory adapted. So again, okay, so how can it be it kind of just blows my mind how the laboratory associated incident was the most likely cause of the first infection, but it wasn't laboratory adapted. So again, there was no leak from the lab. Now we are walking it back. We are now running backwards, trying to face reality, as none of this is publicly available knowledge, and some are unable to make a determination. All intelligence council agencies assess that SARS-CoV-2 was not developed as a biological weapon, despite what RFK says. Yeah. Again, and now more facts of the matter that they have to contend to in this report, that we have no indications that any other researchers were hospitalized because of the symptoms consistent of this of COVID-19. Again, so there were no reports or no people that were sick with COVID-19 from the lab. And the WHO actually did a serology test of these Wuhan Institute of Virology laboratory researchers, and none of them had any SARS-CoV-2 antibodies in their system. So again, there is no scientific data or nothing to corroborate anything with the lab leak conspiracy. Why these federal agencies in the United States are pushing this narrative and why they are putting this false narrative out there, I have no idea. I genuinely have no idea, but they have to contend with facts of the matter. They have to contend with what the reality is, and what we observe is all these things that have happened, all this natural spread, all this natural spillover, all these zoonotic transfers that have happened before. But why would SARS-CoV-2 be different? They've never demonstrated that. No horseshoe bats from North America or South. Oh, okay. Interesting. Okay. So science versus conspiracy. I mean, we really have to think about, this gets into philosophy of science, but I mean, are we ever going to know anything in science with 100% certainty? Like, no, right? I don't think that science could ever be 100% on any experiment. And this has to deal, what we do with science is that we create hypotheses that we can test. And if you're not creating a hypothesis that is testable, then that can be falsified, then you are dealing with a conspiracy. And this was a falsificationism, or critical rationalism, was first brought to the table by Karl Popper. And basically, if you can't test that proposed idea through experiment, then it can be discredited immediately. And these crazy, isn't it a coincidence that, or we just saying, isn't it a coincidence that, like Jon Stewart says, oh, isn't there a coincidence that the Wuhan Institute of Virology is in the same city that this came from? Like, no, it's not a coincidence. It's actually, there's a reason why those institutes of virology are there in the first place. Well, one, it's a major city, and institutes of virology are in lots of different major cities. Two, there is a spillover event that did happen, and there is a predictive nature to science, so that these places that go and try to sample these viruses are put there in these spillover areas on purpose. This isn't a coincidence. So this just saying, isn't it a coincidence that, versus an actual scientific hypothesis, right? Actual scientific hypothesis is, we hypothesize that this virus came from this place via this route, and we're going to test it through blank. In this case, we're going to test it through genetics. Exactly. Correlation is not causation. Exactly. But to give the best case scenario, right? What would actually have to happen in order for this lab leak conspiracy to be true with the data that we have? Because any new theory, any new hypotheses in science has to contend with all the previous data that exists. It has to talk about that. It has to deal with that data. And this is basically what would have to have happened. So you would have to have had a biosafety cabinet, PPE wearing, personal protective gear, Wuhan Institute of Virology worker that had no symptoms, but it was also infected with lineage B, who never sought any type of medical care and had no antibodies ever created in their system. But this person was sick enough with a large enough viral load that they went directly, with no other contact, into the Huanan seafood market, which again has low traffic, so they would have no reason to be there other than to just go there for a certain reason, for who knows why. Now, at this Huanan seafood market, this infected with lineage B person had to spit or cough into the faces and areas of other people in the west and east side of the market, into Siberian weasels, because remember, there's mitochondrial DNA of these weasels and raccoon dogs with RNA SARS-CoV-2 samples. So how are they going to get there other than this person, right? This person is putting enough sample out there that they actually can have a detectable SARS-CoV-2 load, viral load on these weasels and raccoon dogs, and then also proceeds to spit on and cough on these cages, indiscriminately just going around this entire seafood market, coughing and spitting on cages, on animals, on other people, with a large enough viral load that is actually detectable, weeks later, a month later. I mean, it's insanity. And then a week later, by the way, that was just lineage B. A week later, another employee that's working there has to be infected with lineage A now. Remember, it's genetically and distinctly separate than lineage B. So now, again, that same person has to do the exact same thing, and go through and infect everyone, has to go and get the weasels and the dogs and the animal cages. I mean, this is just insanity. There's just the probability of this actually happening is so low. There would have to be an overriding set of data that we have just never could have imagined, or like some ability for all of this to come together that is just completely unknown to us, that would make all of this make sense. I'm not saying that it doesn't exist. I guess it could, but as of right now, this is what science says. We have a fact of the matter. We have the data that we have, and that's what this is how the data makes sense with this conspiracy right now. Or this is what happened. Or we have the natural origins theory, which is just at a Huanan seafood market, you have Siberian weasels and raccoon dogs that have been infected with lineage B. That lineage B came from natural origins, so that could have been a wild animal, that could have been a breeding center that's been infected with lineage B. Now these weasels and raccoon dogs, they're in cages, so there's going to be SARS-CoV-2 samples everywhere inside those cages, and then those animals just transmit through their respiratory mechanisms and infects people on the west and east sides of the market. And these people get infected by walking or buying these animals. And then 1 week later, another separate lineage comes from another animal or another breeding center and infects these cages again, and these dogs and weasels, and basically by buying or walking by these animals again, it allows this virus to actually infect these people. That's the case for natural origins. So those are the two stories that are the most probable right now. So either there is some overriding piece of data or information that makes this all make sense, because right now it just doesn't make any sense. This is just there's just no way that this all happened. But this is what the data says. This is what we know now. Versus natural origins that's happened before, that is what we see with the data, what we see with science, which is what, outside of these intelligence agencies that are putting out this information, I don't know, and I don't know why they would do this. I don't know why they are putting out the information that is so different than what the scientific consensus is. I don't know. I can't tell you. But I can tell you that it just doesn't make any sense right now. What they're saying doesn't make any sense. What the problem is that it impacts us. It impacts us from actually studying what's going on. Because what we need to do is what we need to be studying is how to actually study these viruses more. We need to be sequencing these populations more. And how do we do that? Well, we need international cooperation. So the U.S. needs, U.S. funded virology research in China is very important because now we can actually go and survey these bat populations. Not a coincidence that these Wuhan Institute of Virology was placed there, right? Because just like we had seen with the paper with the 23 different species of bats living in the same spot, we need access to those spaces. And the predictive ability of science gives us the locality that we can actually look at. Large scale sequencing is really important in order to look into these wet markets and the environmental samples to look for deadly mutations that could be accruing in these populations, to look for the next pandemic that could be coming. We need to research which mutations pose these risks. So looking at, we have the sequences, right? The environmental sequences, but now what do those mutations actually mean in terms of having a virus that is deadly? And we need vaccine development, right? That can actually be able to switch and use these environmental sequences to actually switch inside the vaccine. And that's one of the big vaccine, one of the great things about the RNA vaccines is that they have an ability to switch sequences very quickly. And then we also need sociology research on the intersection of this science and culture and these animals so that we can better understand how they interact with one another and how we can impact that. So that is why SARS-CoV-2 is of natural origin. That's just the reality of it. This is the most likely scenario. And until we see otherwise, anyone else is lying to you. So, but that's what I got. Okay. That is an hour and 30 minutes long. So that can't be that. Dang it. I need to cut down significantly. I want it to be like 30 minutes so that people could read it, to watch it. I think I can take out a lot of this stuff. I can take out this, I think. I mean, if I just say it's bat or if I say it's raccoon dog, I mean, does it really matter that I show phylogenetic origins of it? I wonder if the great woolly bat had contact with any of the feeder facilities for the wet market. Yeah, I mean that's so basically those bat populations could interact with those breeding places, and that's one way that they could get introduced. So it probably wasn't at the market that the raccoon dogs were exposed to it. Just because, I mean, we know that some of these organisms are coming in sick, right? And they wouldn't have had the viral load in order to spread it like that. So, yeah, it just doesn't make any sense. I need to put more pictures here. I'm kind of like grasping with the idea of putting this in my notes and then just putting this as like a picture of Karl Popper. So like, for example, I've never done a video like this. I usually normally I would just like sequence. I would just publish that. but I just did. But I am trying to go with something a little different today, or this time, because I really want this. I think it's a really important video. That's a good picture of him. I want a younger picture. I think this is a good one. Okay. Yeah, I do. Yeah, I agree. I know that's kind of like, I my whole point of the channel is to try to like raise people's understanding of like kind of like what's going on, and not just not dumb it down enough so that people don't really understand what's going on, but to kind of like let people learn, but at the same time not talk down to them. I don't want it. The problem with like the PowerPoint slides is that it's very like, it's just tough. It's tough to, you can't really make it engaging. I think I can just remove these and put, well, samples. I think I could like do this. Have kind of like a one sentence explainer on each of them. I think that's okay. I don't want to put too many words on there. Now, these two things, the nitty-gritty details take active cursor. Yeah, I agree. Yeah. I think that's like, I think it's probably better just to take the takeaways from each of these and then just kind of focus on the takeaways. So like, for example, want to see who market very very low foot traffic. So I can take this data and put it into here. Yeah, I think this is just showing basically how this figure came about, but I don't think that's important for people to understand as much. This is the two distinct spreads. Okay, I think that this is actually better than this for showing. This is kind of hard to see these like little dots, so I'm gonna remove that. Spread over time. Okay. If the full lecture could be linked in the description on a separate, less actively monetized YouTube channel, you got some moments worth mining for short clips. Yeah. I honestly, man, I've decided just kind of like bite the bullet on my content, my YouTube content, and not try to like monetize, like not try to be um I just want to make the content that I would want to make. You know, it's one of those things where it's really easy to um get into that game of like, oh, this is what people want to watch, and I need to change things up. And you know, I agree that like mining short clips, like good points of it, is probably a good idea. But if there's a standout moment, then I'll probably do that. But um, the whole goal is to kind of like make the presentation its own thing. It's I don't do this for my living, you know what I mean? So like I'm just doing this for myself and trying to raise the level of discourse where I can kind of like be proud of the stuff that I make is more important to me. Does that make sense? I'd rather be proud of the videos that I make than like putting out stuff that is like so one of the big problems that I think that the reason why there is such a gap that's rising between the layperson and the scientist is that people are dumbing things down too much. People aren't kind of like showing what's actually happening. They're trying to use analogies too much, and they're using those short clips to be like um so they're using those short clips to make those points and to make it like easy understood. But then what's that? What the problem is then it's just the lowest common denominator thing. It's a rush to the bottom where it's trying to make and dumb things down the most versus trying to, which is what I'm trying to do, which is trying to raise the level of discourse, like raise like people's ideas, and so that they can kind of like learn and better understand like the full magnitude of actually what science is actually happening right now, like what people are actually capable of doing. That's way more important to me than being monetized. I know it sounds weird, um, but I ultimately just want to make content that I'm proud of, and I don't think trying to dumb it down. I think that there is like a point to that though. So I am interested in trying to like explain things to like kids. You know, I think that that's the like on this channel, I'm trying to go like kind of full on, full in, mask off. Like this is everything that science can do. You know, take it or leave it kind of thing. But I would be interested in doing like a kids videos or something, you know, like aimed at kids, and there you can kind of you're saying things again. The goal isn't to like pull any punches. The goal is to like speak to them where they are. And my kids like are able to comprehend some really like advanced science concepts because I just tell them about it. I don't just like I don't hold back. I tell them what it is. And I think that that's good. That's what burns trust. Yeah. Oh, thanks, man. Yeah, I want it to be digestible. I don't want it to be like over people's heads. I see some science communication um out there where it's just like it's basically for scientists. It's like science communication to other scientists, which is which is kind of weird. Um because it's like what like the people that if I'm gonna make a Warabi like presentation, for example, that's what this paper is, Warabi. Uh, like I'm not like I have to explain it in a way that like people can understand it. There are people that basically take the Warabi paper and they use all the jargon, and you're like, wait, what a second. Like no scientist is gonna sit down and watch a freaking 30-minute long video about Warabi. You know what I mean about Warabi 2021. They're gonna just watch. They're gonna read the paper themselves. So it's like the audience is so small there. It's just so weird. I don't think jargon is comprehensible. Like I'm trying to explain things in words that are understandable, but not dumbing it down. If that makes any sense. So no jargon, and but if I do use a jargon term, it's for a reason, and I'm trying to explain it afterwards to try to raise the level of understanding. I don't know. The story going into outbreak. Okay. I think this was good. I want to. I like this. I really like this. This Picard paper is going to be really tough for me. This one, probability simulations. I don't know. I think I could probably cut that. I honestly think the Picard paper is only really good for this. If I'm going to be 100% honest, I really don't think that there's any. I hope he's okay with that. This is interesting. I think because the temporal reason, stuff right here. Um, if I can't explain it otherwise, this is such a cool paper. I love it. But man, like explain RDRP and all these kind of like things, that is so tough. You really have to get into it. But I think it's so interesting because it's another line of evidence, right? That's the cool part. It's a lot of slides though. God, 7 slides. And I don't know if it's like that understandable. I think I take this, I put it to my speaker notes like I did before. And I want to do this again. I love this. This diagram is so good. I don't think this is necessary. It just basically shows this information in a different way. You can basically say, oh, bat coronaviruses. But I don't know. Because I don't even explain this right portion at all. All right, fuck, it's 5 o'clock. Oh my God. Well, Anarchist Tech Support, thanks for coming, bro. Thanks for sticking around. I appreciate you. I normally are. This is my YouTube channel. If you want, like, my main content is in here. If you're curious, you wanted to check it out. Yeah, man. I'm gonna be recording this. Yeah, I usually stream later at night and just trying to kind of get things going. I'm usually not streaming on Twitch, so it's actually, I just started doing that a couple days ago. So it's cool that I'm gonna keep doing it. So yeah, Tuesdays at like 5 or 6 p.m. PST. I have, like, there's a panel that I'm usually on. It's called, like, the Ministry of Science. So that's kind of a cool science-based panel that we do, talking about the cool things that's happening in science. But yeah, man. Thanks for coming. I gotta go through this. Oh, I gotta cut it down a ton. I still don't know what to do. All right, it's 5 o'clock. I gotta go. But all right, man. Have a good one. Have a good day. Hope you. Yeah. Thanks for coming again. I appreciate you. All right. Adios.